going to announce the winner of the research group first. The winning topic was Momentum Algorithm for Quantifying Dendritic Cell Movement. Carolyn Crockett and Elizabeth Orrico, Electrical and Computer Engineering, and their project advisor was Scott Acton.
Hi, I'm Elizabeth Orrico. I'm Caroline Crockett. And our thesis focused around the problem of atherosclerosis, which is the #1 killer in the first world. Our work is based off work with biological researchers at La Jolla Institute in California, and they observed cell behavior in mouse arteries that had atherosclerosis that hadn't been observed before, and it was the movement of a particular white blood cell, which is called a dendritic monocyte, and they moved in a way they hadn't seen, and it's called dancing on the spot, and Caroline will tell you more.
So what you see here is our actual data is in 3D, but this is a 2D projection of that data. And in the video of one of the dendritic monocytes, the monocyte moves down and to the right over time, and that's a translational movement. However, the movement that we were asked to quantify is this dancing on the spot movement, where you see the limbs kind of grow, move around, and come back. So given this problem, we started by seeing what was the state of the art, what algorithms are currently out there. And a lot of the algorithms are graph-based methods. And very briefly, these methods involve finding the adjacency matrix of the skeleton, and that just describes the connections in the skeleton. However, there's two problems with this. One is that it's not intuitive. The number you get out doesn't have a physical meaning. And the other problem is that if the graph rotates, the adjacency matrix doesn't change. However, dancing on the spot often involves rotational movement. So these methods really didn't work for us. We turned to designing our own algorithm that we wanted to be much more intuitive for the biological researchers.
So like Caroline was saying, we wanted something that would be intuitive for people to understand movement by both as image processing engineers or as people researching biology at La Jolla. So we turned to like some basic physics concepts to try to think of an idea, and that's how we came up with the idea of momentum. And momentum is the mass of a body times the speed or the velocity of a body equals its momentum, so it kind of gives you a sense of how much energy that particle or body has. So what we did is we applied that concept to each endpoint of our skeleton. So the very first step is get our cell, make it into a skeleton like this, which is the first step. So then we have a skeleton in 3D. This is in 2D, so it is better to visualize. But first we find the mass of each endpoint. And we define mass as the geodesic distance of each endpoint from the graph center. So this red dot is the graph center, and then this one's really close, so 6.7, that one's farther away. Next, we define the distance over time. So here you can see we have 2 frames superimposed over each other, one in gray, one in black. In gray, we use a greedy algorithm to match the endpoints from one frame to the next frame. And then we find the distance between those 2 points. And then since it's from one frame to the next frame, that's over time. So distance over time is kind of the speed. And then so we take the first graph, each point, times the second graph, and then that's the momentum of each point. And then we would take the average of these 5 endpoints, their momentum, and that would be the momentum for that frame.
So in order to test these 2 algorithms, what we did is we created a skeleton simulation. And what this allowed us to do is set the amount of dancing on the spot motion that was present and make sure that our algorithm measured the correct amount. Using this skeleton simulation, we tested both the graph-based methods and our momentum algorithm. So on the right are the graph-based method results, and they have a correlation coefficient of 0.02. Essentially, there's almost no relationship between the amount of motion we set in the simulation and the amount of motion that the algorithm finds. In contrast, the momentum results have a correlation coefficient of 0.85, which is much better. The other really nice thing about the momentum results is we were able to assign physical units. We used essentially an approximation for the density of the dendritic cells and the frame rate to get to grams times micrometers per second. And this is really great for the biological researchers who then compare this unit to, say, translational momentum in the same units.
So our big kind of claim or takeaway is so John D. Bagley was one of the original pioneers of genetic algorithms, and he once said, when man builds machines, he often looks to nature for inspiration, and that was kind of what our team's idea was for this momentum algorithm, is to look to physical concepts to build something that could be understood by us and by biology researchers and hopefully help to find a cure or the cause of atherosclerosis.
Thank you.